I want to start with a scenario that is going to absolutely frustrate a lot of people listening. Imagine you have two neighbors. Neighbor A is doing everything right.
Okay.
By the standard Western rule book. They eat whole grains, they follow the DISH diet, lots of veggies, low salt, and they go for runs 3 times a week.
I can already see where this is going, and it's usually where we say, well, life isn't fair.
Well, it's worse than unfair. It might be biologically rigged. Neighbor A, the quote-unquote healthy one, goes to the doctor and finds out they have high blood sugar, high visceral fat, and are pre-diabetic. Neighbor B is metabolically fine.
Right.
And usually we'd say neighbor A is just incredibly unlucky. But the study we're covering today says, no, neighbor A isn't unlucky. They are just following a user manual that wasn't written for their specific biology.
That is a terrifying premise.
Yeah.
It completely upends the idea of universal health advice.
It really does.
It implies that eating healthy might actually be making you sick if the definition of healthy doesn't match your genes.
We are doing a deep dive today into the HELIOS study. This is coming out of Singapore, and it is a massive deal in the medical world.
A huge deal.
They took 10,004 Asian men and women, specifically Chinese, Indian, and Malay participants, and tracked them meticulously between 2018 and 2022.
And the reason this study is headline news isn't just the size. 10,000 people is a huge cohort. It's the mission. They set out to fix the data gap.
Which is this dirty little secret in medical research, right?
It is. If you look at the vast majority of genomic data, the data we use to design drugs, predict heart attack risk, or create diet plans, it is overwhelmingly based on European populations, Western ancestry.
But Asia holds nearly 5 billion people.
Exactly. So effectively, modern medicine has been using a map of London to try and navigate the streets of Mumbai or Singapore.
It just doesn't work.
It just doesn't work. The HELIOS study was designed to build a new map from scratch. They call it mapping the omni-layer of health.
Omni-layer? That sounds impressive, like a sci-fi concept.
It's essentially a Google Earth approach to the human body. Instead of just looking at the street view, like, how much do you weigh? They peeled back the biological layers one by one.
Right.
They looked at the phenotype. So what we can see, the genome, your DNA, the epigenome, the regulation, the metabolome, and the transcriptome.
Okay, that is a lot of ohms. I want to take this layer by layer, just like the researchers did, because that first layer, the phenotype, is where they found this massive paradox we started with.
Right. This is the hook. They compared the 3 ethnic groups living in Singapore: Chinese, Indian, and Malay.
All in the same environment, basically.
Exactly. Living in the same country, often the same neighborhoods. But the health disparities were stark. The Indian and Malay participants had significantly higher rates of hypertension, obesity, and type 2 diabetes compared to the Chinese participants.
And when you say obesity, they weren't just looking at BMI, were they? They were looking deeper.
Correct. They focused on visceral fat, just to visualize this for everyone.
Yeah.
Subcutaneous fat is the stuff you can pinch on your waist. Visceral fat is hidden deep inside. It wraps around your liver, your pancreas, your intestines. You can be thin on the outside and loaded with visceral fat on the inside.
TOF. Thin outside, fat inside.
Exactly. And visceral fat is the dangerous stuff. It's metabolically active, pumping out inflammatory signals.
So the Indian and Malay groups had much higher levels.
Much higher. But, and here's the mystery, when the researchers looked at their lifestyle data, specifically diet, the Indian participants had the highest scores on the DHH diet.
And just to clarify, the DIH dietary approaches to stop hypertension. That's the gold standard in the West. It's what your doctor tells you to do.
It is. Whole grains, fruits, vegetables, low-fat dairy. On paper, the Indian group was eating the healthiest. They, along with the Malay group, also had higher objective physical activity levels.
So let me make sure I have this straight.
Oh.
They ate better, according to our Western guidelines. They moved more, but they had 3 times the diabetes risk.
That is the Asian paradox.
That is maddening.
It suggests that Western-derived metrics, like the DAS score, might be useless or even misleading for these populations.
So it's not that the participants failed the diet.
No, the diet failed the participant.
They also mentioned the built environment played a role here. It wasn't just about the food on the plate.
No, geography mattered. They found a strong correlation between diabetes rates and the density of hawker centers.
Oh, the food courts.
Those famous food courts in Singapore, right?
Mm-hmm.
And the type of housing. Essentially, if you live in what we call an obesogenic environment, imagine living inside a swamp of cheap, calorie-dense food options, your risk just goes up.
Okay, let me anchor us here before we go deeper.
Sure.
We have a crime scene. the victim, the metabolic health of Indian and Malay populations, the suspects, diet and exercise, they have alibis that were better than average.
Right.
The clue. The standard Western health rules just aren't working.
Perfect summary. So logically, if it's not the lifestyle inputs, the researchers assumed, okay, this must be genetic.
It has to be hard-coded.
It has to be hard-coded. So they moved to layer 2, the genome. And they brought out the heavy artillery whole genome sequencing.
WGS. And that's different from like a 23andMe test, right?
Oh, completely. Those consumer tests usually just spot check for specific typos in known locations. WGS reads every single letter of your DNA book, and they did it at 30x depth.
What does 30x depth mean in practical terms?
Imagine you're proofreading a book. 30x depth means you read every single letter 30 times to be absolutely sure you didn't miss a comma.
Wow, okay.
It's a gold standard for accuracy.
And what did they find in the book?
A lot of new words. They found 252 million variants. Most of them were rare variants found in less than 1% of people. And notably, over 50% of these variants were unique to just one ethnic group.
Which, again, reinforces why a drug developed for a European genetic profile might target a gene variant that doesn't even exist in a Malay person.
Precisely. But here's the plot twist.
Yeah.
They tried to use something called a polygenic risk score, or PRS, to explain the diabetes gap.
We should define that.
We should. Think of a PRS like a credit score for your health.
Okay.
A credit score takes into account your payment history, your debt, all of that, and it spits out one number, 750.
Right.
A polygenic risk score sums up thousands of tiny genetic risks, little typos in your DNA, to predict if you'll get a disease.
And since most of those credit scoring models are built on Western data?
It failed completely. The genetic risk score was basically flat across the Chinese, Indian, and Malay groups.
Wait, flat?
It could only explain about 5% of the excess risk found in the Indian and Malay populations.
Only 5%. So 95% of that massive health disparity, the triple risk of diabetes, is not coming from the standard genetic hardware we know about.
Correct. The hardware, the DNA code itself could not explain why one group was getting sick and the other wasn't.
That is a huge blind spot. So if the hardware is the same, but the machine is running differently, we have to look at the software.
Which brings us to layer 3, the epigenome, specifically something called DNA methylation.
I love the hardware-software analogy. If the genome is the computer tower, epigenetics is the operating system deciding which programs to actually run.
That's a perfect way to view it.
Yeah.
Methylation involves these little chemical tags methyl groups attaching to the DNA strand. They act like volume knobs. They don't change the song. The DNA code remains the same, but they can turn the volume all the way up so the gene is screaming, or hit mute so the gene is silent.
And this is where they found the difference.
Big time. They identified over 16,000 specific spots, they're called CPG sites, where the methylation differed significantly between the ethnicities. The software was configured completely differently.
Did they find a smoking gun gene in the software settings?
They did. They found major disturbances around a gene called PLAG1.
What does it do?
Think of PLAG1 as a foreman. It's a transcription factor, so it bosses other genes around, and is heavily linked to how the pancreas develops and how it secretes insulin.
And since type 2 diabetes is essentially a breakdown of the insulin system,
Finding a glitch in the foreman gene is huge. The study showed that the volume knob on PLAG1 was set to a position that predicts future diabetes. But here's the most empowering, or maybe frightening part of epigenetics. These volume knobs aren't fixed at birth.
they can move.
They are fluid. The study found that these methylation patterns correlated with diet quality, adiposity, so how much fat you have, and even education level.
That's wild. So things like your education or the neighborhood you live in are literally leaving fingerprints on your DNA regulation.
Yes. Social factors become biological factors. Your environment reaches into your cells and turns the volume knob on the PLAG1 gene, which then alters your insulin production.
So that's where the missing 95% of risk is hiding.
That's the theory. It's not in the code. It's in the regulation of the code.
So to pause and make sure I've got the narrative arc here. 1, the diet scores didn't explain the sickness.
Right.
2, the raw DNA code, the hardware, didn't explain it either.
No.
But 3, the regulation of that DNA, the software driven by the environment, showed massive differences.
You've got it. But we can go deeper. We've looked at the code and the software. Now let's look at the output, the metabolome.
This is the blood work on steroids.
Basically, metabolomics studies metabolites, the small molecules left behind by metabolism. Imagine checking the exhaust pipe of a car.
Okay.
By analyzing the smoke, you can tell if the engine is burning oil, if the fuel mix is rich, if the catalytic converter's broken.
So they checked the exhaust of these 10,000 participants.
They measured over 1,000 of these molecules. And this is where we get to what I think is the most concrete example of precision nutrition in the whole study, the chapati metabolite.
This sounds like a detective story title. The case of the chapati metabolite.
It really is. So they found a specific lipid, a fat molecule called 1-margaroyl-2-arachidoyl GPC.
I'm definitely just going to call that the GPC lipid.
Good call. They found this GPC lipid was way higher in the Indian participants. When they traced it back, they found it was strongly associated with eating red meat, dairy, and specifically chapati.
Which is a staple whole wheat flatbread in Indian communities.
Exactly.
Okay, but millions of people eat whole wheat. Why does it matter here?
Because of a gene by diet interaction. The level of this lipid isn't just about how much chapati you eat. It's controlled by a gene cluster called FADS1 and FADS2.
FADS, fatty acid desaturase.
Right. These genes control how your body processes fats. And the Indian population has a high frequency of a specific FADS1 variant, a specific version of that gene.
So you have a unique genetic variant.
Combined with a cultural staple food, chapati. That combination creates a unique metabolic byproduct.
Which explains why the DSH diet fails. If you tell everyone, eat whole grains, but one group has a FADS1 variant that processes those grains into a risky lipid.
You're technically giving healthy advice, but biologically you're causing harm. That GPC lipid was inversely associated with heart health. This is the definition of precision nutrition.
So the question isn't, is bread healthy?
It's, is bread healthy for you given your FADS1 status?
That changes everything. It's not just about eating clean, it's about chemical compatibility.
Precisely. And they found similar mechanisms in the final layer, the transcriptome.
Layer 5, this is the RNA.
Right. If DNA is the cookbook in the library, RNA is the handwritten copy of the recipe that the chef is actually using in the kitchen right now.
So it tells you what's on.
It tells us what genes are effectively on. They found almost 3,000 genes with different activity levels across the ethnicities. And a lot of them were linked to inflammation and, interestingly, COVID-19 susceptibility.
Which tracks with what we saw during the pandemic, where different ethnic groups had vastly different outcomes.
Exactly. But they honed in on one specific gene called TXNIP.
TXNIP, what's its job?
It is a major regulator of how cells eat glucose. If TXNIP is off, your cells don't pull sugar out of the blood effectively, your blood sugar spikes, and you're on the road to diabetes.
And let me guess, the volume knob for this gene was different.
They found specific dimmer switches. The technical term is cis-EQTLs.
Okay, cis-EQTLs. Let's break that down because it sounds dense.
Sure. Think of a gene as a light bulb in the center of a room. A cis-EQTL is the dimmer switch located on the wall right next to that light bulb.
So it's local. It's not a master switch somewhere else in the house.
Exactly. It's cis. It's right there. And it controls the brightness of that specific bulb. The HELIOS study found new cis-EQTLs for TXNIP and other genes that are not in Western databases.
Wait, so Western science didn't even know these dimmer switches existed?
Because we weren't looking at Asian DNA.
Yeah.
If you only study European houses, you assume all light switches are on the left wall. Then you go to an Asian house, and the switch is on the right.
We missed a primary mechanism for diabetes because we were looking in the wrong place.
We were trying to fix the light bulb without realizing we couldn't even find the switch.
That is mind-blowing. Now, before we wrap up, how do they know this is real? I mean, finding a correlation is one thing. Proving it causes disease in the real world is another.
This is the big brother bonus of Singapore. They use something called a trust platform. They link the research IDs of these 10,004 people to their national medical records.
So they have their entire medical history.
20 years of it. 1.6 million lab tests, prescriptions, diagnosis codes. They could look backward and forward in time.
Why is that linkage so important?
Because it validates the biology.
Yeah.
If the epigenetic data said, hey, this person has the Cleage-1 methylation pattern that looks risky, they didn't have to guess.
They could check the records.
They could check the records and see, yep, 3 years after this blood draw, this person was prescribed metformin for diabetes.
That proves the predictive power. It takes it from theory to clinical reality.
It is the gold standard of validation.
So pulling this all together, we've gone from the macro people eating salad and getting diabetes to the methylation of the Cleage-1 gene, the FADS1 interaction with chapati, and these missing dimmer switches on TXNIP. What's the big takeaway?
The takeaway is that Asian is not a biological category. It's too broad. And standard care, based on European data is insufficient. We are moving toward a world where your doctor doesn't just ask, how much do you exercise?
Right. They need to be looking at your ancestry, your methylation profile, and your metabolites before they tell you what to eat.
Exactly. This is the dawn of true precision medicine.
Yeah.
And the next step is SG100K. They're expanding this study to 100,000 people.
Wow.
That will give them the statistical power to find even rarer variants and more subtle interactions.
It's exciting, but it also makes you question a lot of current wisdom. It
Yeah.
feels like we are in a transition period where the old rules are breaking, but the new rules aren't quite printed yet.
We are.
Right.
We have to realize the map we were using was incomplete. But studies like HELIOS are finally filling in the blank spots.
Here's a thought to leave you with. We talked about the -H diet, a universally healthy diet, failing a specific population because of their metabolic profile. It makes you wonder, what universal health rule are you following right now? Whether it's intermittent fasting, high protein, low fat, that might actually be wrong for your specific biology.
That is the question.
Yeah.
And until we all have our genomes and metabolisms sequenced, the best advice might be to listen to your body's data, your blood work, your energy levels, more than the bestseller list. If a healthy food makes you feel terrible, trust your body over the book.
Stay curious about your own biology, everyone. Thanks for diving deep with us.
Thank you.